If you think about research in humans, it means if you want to bring new ideas in the market, you will need to investigate in bench tests, then in animal studies, and later in the humans. This can take $1 billion of costs and take easily 5 years and more. Our idea of the SIMCor project is to use computer technology to make this whole thing shorter and safer for the humans, and also to overcome the problems of the animal studies.
An In-Silico clinical trial is a virtual representation or virtual variant of a real clinical trial, which is there to assess safety, efficacy, and conformity of medical devices. In SIMCor, we want to use In-Silico clinical trials to assess the safety and efficacy of our 2 use cases, PAPS and TAVI. And for this, we use different physics-based and data-driven models to model device implantation and device effect. We also use virtual cohorts to artificially increase our sample sizes and to allow assessment in large numbers of virtual patients. And using these technologies in combination, we therefore can provide evidence of whether the devices are safe to use in patients, whether specific risk factors have to be considered, or we can also provide guidance in the redesign or in the update of designs of these 2 use cases.
SIMCor platform is intended as an open environment for collaborative research and development among device manufacturers, medical authorities, researchers, and regulatory bodies. SIMCor platform is structured in 4 main components. The first component is VRE Drive. It is a cloud storage server where we are storing the project data. Then we have the second component, which is the virtual cohort generator, providing a set of tools to generate a virtual population of patients based on some given characteristic as input parameters. The results are used to simulate the device implantation. We also have a component for doing clinical trials called our statistical environment.
One of the most interesting aspects of SIMCor is that we try to reuse data which were routinely collected in our clinical routine in order to acquire anatomical and functional information of the population of interest for our projects. We based in particular to CT images to reconstruct the implantation site of our patients, and then echo pictures to understand the functional. And these were essential to create the virtual population for our group.
A cohort, if you speak about patients, that are a lot of patients. And a virtual cohort is that we actually have a lot of virtual patients. Now, what do I consider as a virtual patient? I consider as a virtual patient a mathematical model based on physics or physiology that represents or mimics a patient. So we are able to simulate the physiology. So, for example, we have a TAVI device for heart valve replacements, and we can simulate the situation before and the simulation after for one patient. If we are able to make such a model and we demonstrate that it's realistic for, let's say, 100 patients, then we can generate by interpolation thousands of new patients by varying the parameters. And then we have to check, of course, if they're real. So we have to apply a validation strategy. And to validate that, we have 3 validation steps within SIMCor. And the first validation step is on patient level. So we really want to mimic one-to-one real patients. And we demonstrate that our model is able to realistically mimic the physiology or the pathophysiology of that individual patient. Then we do the variation, and we generate thousands of them, and we select the ones that are good. And we do that based on our acceptance criteria and on also the data set that we have. And we do that on a completely independent cohort. So what we actually do, we have a patient level validated model. We vary the parameters, we have acceptance criteria, we then look to the distribution of the real world data that we also get that distribution. So we call that the self-validation, and then we do a cross-validation where we try to see if our set model and our set filter criteria also create virtual patients that mimic a completely independent but clinically matched cohort. And that's what we call the cross-validation strategy.
Let's define first what is an in-silico simulation in terms of TAVI. What we want to do is to perform a calculation of the blood flow through a TAVI. A TAVI is an artificial valve, and when the blood rushes through the valve, there comes to, for example, recirculation, so the kinds of vortices of the blood, and these vortices could be a reason, for example, for thrombosis or other post-operative complications. And our aim is to look what kind of implantation, for example, is needed to reduce the risk of thrombosis, and what a device should look like to reduce thrombosis or even do not have the thrombosis.
We developed a numerical model based on the finite element method, in which we can simulate the deployment of a TAVR stent. And using this in a virtual patient cohort, we can study the effect of different deployment strategies, stent sizes, and for example also patient-specific features of the aortic root geometry on the final shape of the stent, but also on the forces exerted on the tissue and the stresses induced into the tissue. And we can then use these finite element simulations to gain more insight into what patient-specific features contribute to post-operative problems in real patients. And eventually the aim is that clinicians can use this knowledge to optimize and plan the procedure and hopefully help prevent post-operative problems in real patients in the future.
For the project, we analyzed the tissue properties of animal and human tissue, and we used this to implement the data into a constitutive model to describe the vessel wall. And for this, we needed first to collect tissue samples from animal, but also from humans. After collecting, we did biaxial extension tests, where you mechanical load those tissue samples in two directions, which mimics quite closely the in vivo conditions in the body. And we did this for two arteries in general for the project, so the ascending aorta and different regions of the pulmonary artery. And the second part was the microstructural investigations. So we wanted to figure out how the collagen fibers in the arteries we are investigating look like. So the alignment and dispersion of collagen fibers, which are the main load-bearing constituents. For this, we used a multiphoton microscopy. And based on the data from the mechanical testing, but also from imaging, we then identified the material parameters of a constitutive model, which can describe or mimic the anisotropic response for those specific arteries. And this constitutive model was then applied afterwards or used afterwards in more sophisticated computational simulations to simulate or model the migration and deployment of the used medical devices.
The pulmonary artery pressure sensor is an implant which is implanted in the pulmonary artery of a heart failure patient and can pose a serious risk to the patient if something happens like perforation of the vessel or migration of the sensor or even thrombosis. And we try to assess these risks beforehand using these in-silico models. And for this, we develop models to simulate the interaction between the device, the anchoring, the fixation mechanisms of the device with the vessel wall, and also the interaction of the body of the implant with the blood flow. And we validated these models in terms of engineering properties. And when we then finally have the validated models, we can use them to infer the behavior of this implant in a human population. So then we can make this transformation or this translation from an animal trial towards the human trial.
In-Silico trials is an important mechanism for regulatory evaluation of medical devices or pharmaceutical products. To be able to deliver such kind of strong contribution for regulatory evaluation, In-Silico clinical trials needs to have standardized workflows, but also a lot of handbook that people could replicate. That's where the standard operating procedures or SOPs play a big role to make things easy, but also comprehensive, such that all the steps of the In-Silico workflow are covered.
What are the potential benefits of in-silico methods in the area of cardiology? The principal idea is to complement and to optimize studies with humans, meaning clinical trials. Recruitment is difficult, it's extremely expensive, it lasts very long, and any help to improve that process would be of tremendous help with respect to reducing costs, shorten the regulatory processes for medical devices, and also it would shorten the time to come to market.
Technologies, especially with respect to socioeconomic effects, is a rather new topic. Therefore, we use the qualitative approach to better understand all the implications of in-silico technologies on 4 different levels. The firm, so what changes in the production process, the product development cycle, the market for medical devices, the healthcare system, and society as a whole. And what we found is that in-silico technologies have a great potential to change the dynamics of the product development, and therefore have the potential to, on the one hand, decrease costs of production, but also to increase safety for the patients and to also reduce animal trials, although all of this rather depends on the regulatory framework that, for example, the European Union or the US government imposes on in-silico technologies.
There is a lot of work that has been going on for decades in the field of in-silico medicine. However, there is still lack of standardization of models and methodologies, and this does not allow for its acceptance by regulatory bodies for medical device testing and approval. So what we have done was to try to gather feedback from clinicians, from industry, from regulatory bodies and patients as well. And also we have tried to join forces with the other EU-funded projects in the field with initiatives like the InnovaHeart workshops to try to overcome together the open technical issues in the field and have a unified voice towards regulatory bodies to call for the acceptance of in-silico methods for device approval.
Our strong belief is that with our technology, one day we will not need any animal studies anymore, and that we can reduce also human trials to a minimum, that we can invent devices made for men, women and children, and by that that we can really also decrease the time from the idea to a product to a very short period. So if we achieve that, that would be really our long-term goal.